Hello, everyone. We will be starting the meeting in a couple of minutes. Thank you for your patience. Hello, everyone. Welcome to the webinar. The webinar will start in 5 more minutes. In the meantime, you can introduce yourself in the chat section and get to know the participants. Hello, am I audible?
Yes, Grigori. Yes.
Yes, you are.
All right, thank you. Let's start the webinar. Hello, everyone. A very warm greeting to one and all. My name is Srigavi Krishnakumar, and I'm a research intern at Fine Biotech. I would also be the host for the webinar for today. So to begin with, I would like to welcome you all to today's webinar on OmicsLogic Bioinformatics and Data Science Research Fellowship Program. Before getting into the further details, I would like to briefly talk about the program. The Research Fellowship Program has been designed to help young researchers and students take advantage of the bioinformatics resources for analysis of complex life science data and become well-versed in bioinformatics. The research fellows participate in cutting-edge bioinformatics research led by expert mentors. The fellowship program will offer a combination of online resources and mentor guidance to prepare you and help you complete a bioinformatics project. The registration fees will cover the cost of training, mentorship, and big data server. The platform is a cloud-based analytical server used by research labs and independent scientists around the world. So before we begin with today's webinar, let me introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. The OmicsLogic training has been completed by over 24,000 participants from 187 countries in over 300 workshops. Due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. The OmicsLogic program follows a project-based learning approach using research-grade tools to analyze data from top peer-reviewed journals. The OmicsLogic training has been completed by participants from 162 countries in over 300 workshops for 6 different specialization tracks, ranging from oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. As a result, we have acquired professional certification to offer our curriculum and support research projects using our OmicsLogic project-based curriculum. Together with our existing modules for which you will gain access to the OmicsLogic Portal, where you can try and explore these modules, exercises in pipeline, building, coding, and various methods of analysis using project sources from peer-reviewed publications. Today, we will discuss about the research specialization tracks, program structure, outcomes of the program, take your queries, and listen to our research fellows projects. Also, we will take you through the program resources and help you get started with your training and research journey. So first of all, let's begin by signing up on the OmicsLogic Learn portal. To do this, you can visit this website. I'm sharing the link in the chat box. So once everyone has the link open and ready, we can continue. Please comment yes so that I can proceed. Okay, great. So let's begin. So once you come to the website, you will see on the top right corner, the tab that says login. Click on that one. If you already have an existing account, please sign into your account. Otherwise, we'll create an account. So to create an account, you will be asked to enter 3 main details: your name, your email ID, and your password. Or you also have an option to join using your social media handles. That includes your Google account, Facebook account, your Apple ID, GitHub account, and your Twitter. So I'll pause for a while. Once you create an account and log in, please let me know and we can log in together. All right, so since I already have an account created, I'm going to sign in using my existing email ID and password. Once you log in, you should be seeing this the number of courses that are offered on the OmicsLogic Portal. And on the top right corner, you should be seeing a tab that says Welcome Back with your email ID. Click on that tab, and you should be redirected to the profile setting page. Here you should see an option to upload your profile picture, link your social media handles, as well as provide a brief description about yourself. And this way, the like-minded researchers and students in the field can get in touch with you and connect with you. All right. So once we have this, I'll like to invite Dr. Raghvan and Lakshmi Narayan, who will be talking about the program structure, specialization fields with example research fellow projects. To give a brief background about Dr. Raghvan and Lakshmi Narayan, he's an accomplished computational biologist with more than 4 years of research experience in subject areas like multi-omics data analysis and data science methods in precision oncology and protein or DNA structural bioinformatics. With a strong background in bioinformatics and biophysics, he earned his PhD in computational biology from the Max Planck Institute, Germany. So please, I would like to invite Dr. Raghvendran sir for explaining the further details of the project.
Thank you. Thank you very much, Sri Gauri. Let me find ways to share my screen. Okay. Okay, I think we should work on steps. Can you see my screen? I think it's still working on this.
Yes, it's visible.
Okay, thank you very much, Sri Gauri, for a very nice introduction. You don't have to solve me. So I also got confirmation that participants can see my screen. And I still cannot see my screen. It's that. Okay. I think both you and me, both of us can see my screen. So welcome everybody to this. Welcome everybody to this webinar. And I am Raghavendran. I'm a research consultant and bioinformatics mentor with Pine Biotech. And I will be your mentor throughout your research fellowship program. So I'm very excited to share with you how this program works, and we will answer some specific questions related to what is this program about and how to join the program, how to submit a project on OmicsLogic, and what are the general structure of the program that will help you perform your own bioinformatics research with us. So this program can be roughly divided into 3 main stages. So at the first stage is a training stage. So this is a stage which sets the direction for participants or students like you to define in which area or in which research field that you are going to work on. So some of you might have a very clear idea on which field or which research topic that you want to work on. And some of you might have also had some introduction to bioinformatics as a course or as a curriculum or as a different introduction means. So, but some of you also would like to gain more knowledge before making a good decision. So for those who are still deciding and who still are not from a bioinformatics background, we always suggest you to start with two very basic courses, that is Introduction to Bioinformatics and Bytes and Molecules. So these basic courses are designed to introduce the basic concepts of bioinformatics and its application to different research fields or areas that employs various omics data types, which can be subjected to multitude of analysis methods. So we are introducing you several methods. So by the end of these 2 introductory courses, you should be able to understand what are the different methods employed for different omics data types. And also, you also should have a clear idea of in which field or in which data type or in what question that you want to work on. Not question, at least a focus on which direction that you want to move. So at the end of the first stage, you should be able to, for example, have all the necessary skill sets to identify a specific research question that you would want to work on. So in this stage where you are going to identify a research topic, so you will have to first find a data set that has the potential to answer your research question. So more on this a little later. So in the next stage is where you actively pursue your research, discussing results, inferences, and all the necessary suggestions and feedbacks from mentors all along, right through. So after these three very important phases, you will have to prepare a presentation that you will submit in our portal, the OmicsLogic Portal, as a project, so that you can showcase this project and share with your network of peers and share your poster if you have prepared in conferences and in meetings, scientific meetings that you can attend. So let's again go through for example, sorry, I think it's not stopping at one point. Yeah, so we throughout the course of this program, we have a weekly progress report form that you will have to fill up and submit to us, and you will have to discuss your progress with the mentor at least once in a week. So these progress reports are excellent way to evaluate how you progress through the research fellowship program. It is helpful for you and also helpful for us to find or to place you where you are currently, and then to help you in at all the necessary steps of your research progress. So our learning resources for this program includes various courses that cover different specialization fields. So we have specialization fields like that focuses on the bioinformatics parts of the omics field or omics data type that you would want to start with. Like if you want to work with metagenomics, then you can start with metagenomics courses. If you want to work with transcriptomics, you can start with transcriptomic course. That both of these courses and also other ones goes in depth in the type of data that you are going to analyze and the various different options of analysis methods that you will employ for different questions and different answers that you are pursuing from these data types. So you can also decide on different data science skill sets that you want to start developing so that you can apply to various omics data type in general. So for those who would want to go through skill set development fields or specialization, we have multiple coursework that explains or that introduces you to Python environment and R environment and different data science applications and machine learning, which includes machine learning and deep learning methods. So all of these methods can be generally applied to various data types that one can obtain from several analysis methods that are designed specifically for different omics data types. So one example of how these courses are designed, let's take a look at designing a bioinformatics research project courses. So these courses are designed to take you deeper into those specific respective topics in a step-by-step process that are outlined into separate lessons. So for example, this is a course which we suggest to all the participants so that they can understand the necessary steps involved in how to design a bioinformatics project. So more on this a little later. So also, throughout these courseworks, we have several practical assignments along the coursework and example projects that takes data from publicly available resources and the data that are used in popular research articles. And we will design separate analysis pipelines or separate analysis methods that is going to answer a research question that is not straight, that is not related to what was the focus of the research paper. So we will take the data and then try to combine different analysis methods in employing or in using these publicly available resources to answer the research question on our own. So while you are going through these example projects, you will get a good understanding of how to take a publicly available data and how to create your own analysis methods that is going to answer a specific question or a hypothesis that you will form from basically understanding some introduction about the data as described in the associated paper or publication. So these different exemplar projects are designed to include variety of topics like in epigenetics, human health, agriculture, virology, immunology, precision medicine, etc., etc. A lot of different ones. So also microbiome also. So these practical exercises and these projects will give you necessary exposure to understand the data-driven discovery of real research behind these projects. And it will also encourage you to think critically about how to go about designing your own projects and what are the necessary ingredients in creating a successful bioinformatics project. So as I mentioned, the basic purpose or the fundamental purpose of doing these practical assignments is to get a deep understanding of data specific challenges and also to showcase how various methods of analysis are applied in real world situations. So when you compare the results that you obtain with the one that are focus of the publication, then you can clearly understand how the questions are formulated to get to the publication level from such a publicly available data or such a vast quantity of data that is dealt here. So this can be your project at the end of this analysis also. So once you have acquired all the necessary skills and you are performing your research in a good healthy progress, and then you can use these skills to apply them to various different research problems that shapes the bioinformatics and biological research of today. So it can be from precision medicine, it can be from population health, can even be space and astrobiology. So we have various example projects that that are categorized under such different categories. So it will give you all the necessary introduction to take the data from these specific fields and then apply your own analysis methods for the question that you are interested in trying to ask from those data. So let's start going through how would you find a research topic. So let's take a practical approach to how to get or how to identify a research topic. So as we said, the major steps that are important to design a research problem that we have been discussing so far is first you have to frame the problem, and then to frame the problem you have to understand what is the latest current trend in that specific problem. So you can, by framing the problem, like you can try to narrow down that I want to work on cancer, or you want to be even more specific. I want to work on, let's say, head and neck cancer. And then you can understand the literature, or you can go through the literature, review the literature, the scientific results for the last 5 years or even last 10 years on this specific topic. So once you have gone through the literature, you can then identify that specific gap. So the literature review should tell you what are the areas that are addressed more frequently or more in a very detailed manner. And also it should also give you a hint on what are the areas that are not covered generally. So you can try to approach that area or try to take that specific area and then collect a raw data that will, or you can actually find publicly available resources, data that is necessary to answer your question or your problem or answer the gap in literature that you have identified so far. So once you have collected these data, then you have to create a processes or create a sequence or create a pipeline for the analysis, detailed analysis of this data. And then you can, you start by exploring the data to understand the patterns, and then you perform in-depth analysis that you have designed for the data. And once your analysis is complete, then you infer from the results and then perform necessary biological implications by relating it to the characteristics of the sample or to the nature of the problem that you started with. So for example, in head and neck cancer that I started with, I might have started with trying to categorize the tumor tissues. So if I can collect the necessary data that will help me in categorizing or classifying the tumor tissues into multiple categories, then I have to find whether my analysis substantiates this claim or substantiates this hypothesis. And once my analysis and my inferences are complete, then I can put together this as a story and communicate the results to the peers. So let's start how to find a topic or how to choose a topic. So you always start with topics that you know about or topics that you care about, and etc. For example, this could be a personal reason, or this could be a reason that you are deeply interested in. So you can start by noting down, or actually by collecting few slides or few information about the background, about the topic background. So and then you have to start preparing or start spending few time in analyzing or researching the topic. So how much has been known? So what has been studied, and among those what is studied, what is left to be studied, and what will be interesting in this topic that will be interesting to the audience, and that will be interesting to you also. So you have to do something that you also care about. And then once this is narrowed down, and then you have to find a data that is going to answer the question that you can choose from this in this topic. For example, if the topic is very specific or very narrowed down, you might not find sufficiently studied data or sufficient volume of data that will answer your query. Or if the topic is too wide or too broad, you might find a lot of data, and you will be left with a lot of choices to make a good decision. So you have to find the right balance that that will maximize, or that will help you in identifying the data, perfect data that will answer the query that you have in your mind. So how to do this? For example, so finding right questions or right sources are one of the issues. So this is always a back and forth process where, for example, in the original research, you take the raw data and then you perform the analysis, and then you compile different analysis, and then you find a solution. Or you can also go back, you can take from the publicly available resources and collected data, compiled databases, and then you explore for the possible trends. And then from these trends, you can actually design a question, whether the initial question that I have in mind can be substantiated by following some of the trends. So we will get an example for this in a while. So for example, if you to narrow down on the topic of research identification or topic identification, you first go through the reviews, and then you can categorize those reviews on their analysis methods or different questions that they are answering. And then once you have a thorough meta-analysis done or thorough analysis done on the reviews that are available, or a publication that have done for the last, let's say, 2 or 5 years of the data, then for your topic, then you can identify the unanswered questions, and you can identify the gaps, and you can also identify some of the debates and controversies which are left unanswered in this topic. So these are the areas which you need to focus on. I mean, on the areas which are obviously answered by many people, you don't want to reinvent the wheel. So we want to identify those areas which are not covered a lot, and which has a lot of potential to improve the science, or which has a lot of potential to fill the gaps in the science in that particular research area. So that gap, and how to answer, how to design specific analysis methods to answer those gaps or answer those unanswered questions, will define your hypothesis or question. So you always start with trying to find a topic. So these are often used, or the resources that are often used are NCBI, repositories like PubMed and bioprojects and gene expression omnibus. These are all the and SRA. These are all the repositories where you will find publicly available data. So you can start with PubMed and then look for or search for some of the topics that excites you. And for example, let's say that if I want to work on SARS, so obviously SARS and coronavirus, these topics are famous for the last 2 or 3 years in the pandemic era. So once you understand different questions and different reviews that are done on SARS, maybe you will find a very interesting topic. So let's say this topic of understanding the imbalanced host response to coronavirus infection, so is the one that excites you. So what do you do? So you start to find data sets that are either associated with this publication or a data set that are associated with host response to SARS infection. So if you find data sets that are associated with this publication, then you can explore the data set for possible trend, and that will design your query or design your question. Or you can, as I mentioned, generally search in the databases mentioned above, like Gene Expression Omnibus or SRA or bioproject databases for host response or human host response for SARS-CoV-2 infection. So this paper has a data set associated with that, so we can see the accession number, the GUIDs, so we can get through the GUID. So this data set in the Gene Expression Omnibus data page will give you some basic information about what the data is all about, and what was the study design, and what was studied, and what are the samples involved, and what are the different replicates of the samples that we can find. So, and this also gives you access, or this also gives access to processed data. So we are not going to perform a detailed analysis of the processed data, but the processed data is an excellent place for you to start looking for the trends that will answer your queries. So when you take the processed data and then quickly perform a principal component analysis to study the sample distribution across the transcriptomics data in this case, then you can, if you can find such trends which can clearly separate samples that are of control type and samples that are given or that are subjected to treatment from RVA and RVC. So this trend gives you a lot of promise that you can specifically focus on, for example, responses due to RVA and responses due to RVC when compared to the control samples. So you have to perform such a basic exploratory analysis on the processed data that is available on almost all of the GEO data sets. So this could be a back and forth process. Some of the data set might not be conducive for a linear analysis like PCA, and some of the data set could be large, and you have to go through this process for a couple of more than one data set. That's what I meant. So this trend is promising. So when we see such a promising trend, so we collect the raw data that is also linked to the SRA, or short read assembly database, from this GEO database. All of these are from the one umbrella data repository, NCPI, database with NCPI. So we collect the raw data, FASTQ data, and then perform our own analysis or design our own analysis process for the raw data. So why do we do that? We can, for example, check if difference in a couple of analysis algorithms that we choose will change the data, or maybe we can take the raw data and perform our own normalization or processing or different processing methods. And most of the processing downstream analysis methods that you are going to employ will require the raw output from such a pipeline which is a sequence of analysis that you employ to the FASTQ or high throughput sequencing output. So we pre-process the data and then we map it onto the reference genome, for example, and we quantify the mRNA molecules that are mapped onto different genes. So this is a very short step-by-step process of transcriptomics analysis. This is just one example of variety of different analysis that you can perform on omics data. So this will actually give you or this can actually result in a downstream analysis from which we can learn what are the genes that are differentially expressed when you compare two conditions. For example, in here, treatment through one rotavirus strain or treatment to the other rotavirus strain compared to the control samples. So you can design your own analysis method that is different from the one that is published, or you can use an updated version of the human genome reference. So you have a variety of different reasons to perform your own analysis methods. So once you perform these analysis and get hold of structured data, then you can start exploring the data for the possible patterns and well-defined patterns. We already had a hint of the trend that could be explored from the data, and then these pattern exploration or these data exploration will tell you the necessary transformation or normalization that you need to perform so that you can use variety of different downstream analysis that will validate your hypothesis, or that will throw light on the question that you are working with. So yeah, so then you can perform in-depth analysis on the structured output. You can perform, for example, you can identify different genes that are differentially expressed, and then from the genes you can identify the pathways that are enriched, or you can identify which specific network, that is biological pathways and network, that is network of genes that is involved in characterizing this response, for example. And then we can also study various different biology of the sample, like if your data involves response to a drug resistance or treatment resistance, then we can also study the basic behind this development of this resistance from this analysis, for example. And at last, you will have to, after performing the data analysis and after performing the biological implications, you have to string all of these in one good story or one good project that will link what you have done and how you have done it, and what you infer the most from the variety of different analysis that you have performed for the project. So yeah, this, and then once you have completed or compiled the project report, then you can publish that in our Learn OmicsLogic portal, and and that will be public to many people, and you can share it with your peers. And then you can also, if you create a poster like this, you can also share those posters in the symposium or in several meetings that you are going to attend. So next up, here are some of the latest or some of the projects that are developed in our platform, and which are part of the research fellowship program from various different specializations. So we have, for example, in this case, this is a poster for a project done in collaboration between Noguchi Memorial Institute for Medical Research, and this poster is on malarial parasite Plasmodium falciparum, and its antigen, which is an antigen with a rotary neck protein binding binding. So the interactions study. So this interaction is extremely critical for this for the falciparum to invade into host human response, host red blood cells. So different malarial vaccines are actually that are known to prevent such binding and to prevent this interaction, and these different candidates or these different vaccines have variety or variable success or variable successes at different stages. So this study actually performs a haplotype analysis or network analysis of genomic sequence data from the variable infection rates, and it compares with the data that is from the repository from another region. So by mapping on to the genomic variants that one identified from this haplotype study onto the protein structures, this paper or this poster actually concludes that there are some local variations and there are some epitope affecting variations in geographical locations that are different for different malarial parasites, and that needs to be taken into consideration while developing vaccines for malaria in a bulk. So and also you can also publish your results in bioRxiv preprint. Then you have a paper or almost a near publication ready output that is available in the public, and you can link it to your ORCID ID, and then you can also share it with your share it with your peers, or use it for your future endeavors. And yeah, bioRxiv is one of the best way to store your almost ready projects. So there are other ways to do this also. So you can also develop your profile, and then this is from Sina Dolaner, and who finished her projects and she was able to work on leukemia and identify differentially expressed genes and protein coding genes that are involved in biogenesis and of these leukemia cells. And she also identified several non-coding RNAs and that are found to be significantly expressed, and this culminated into a research paper. So you can also publish your research if your research fellowship yields very promising results, or if this could set the stage for further analysis that could result in a very promising paper. So yeah, with that, I'm open to some questions, and I pass the stage back to Sri Gauri.
Thank you so much, sir. That was really informative. If the participants have any queries or technical challenges so far, please mention them in the chat box and we can help resolve it. I'll wait for a couple of seconds. All right, so next I would like to invite Mr. Ilya Bronski. He is one of the co-founders and CEO of Pine Biotech. His background is in applied mathematics and data science. He has facilitated and managed collaboration with researchers at Stanford, UCSF, Louisiana State University, and other academic and pharma R&D teams. Mr. Bronski also oversees the strategic development of research projects and manages the team responsible for bioinformatic analysis, training, and collaborations for Pine Biotech. In this part of the webinar, he will be discussing about the research fellowship outcomes in the US. Over to you, Mr. Ilya Bronski.
All right, thank you, Sri Gauri, and thank you, Raghav, for giving a good overview of the program itself. I briefly want to mention that, from a perspective of someone that hires people with bioinformatics backgrounds, I think it's important to understand, as a student, potentially you might not really know what that work might look like, what the projects and what the different challenges might be. And from the employer's perspective, and the employer here could be either in a company or in a lab, a research lab, a PI of a research lab, they don't really know how to evaluate someone who's coming to them and trying to show their resume, or trying to, come as someone who knows, let's say, bioinformatics or even biotechnology for that matter. So in a lot of cases, when you're faced with that critical interaction, what are some of the things that you actually want to be able to demonstrate? One of the things that will always come up is the soft skills, so to say, right? So how well can you present yourself? How well can you answer questions, or maybe think on the spot about some question that involves maybe a non-direct answer, right? Where you have to show your ability to think, consider the different options, come up with maybe some kind of a decision that needs to be made. And because these kinds of interactions can never fulfill the role of getting to know someone, right? The interactions are very brief and non-conclusive. That's why your resume is not enough. That's why just coming to the interview is not enough. In a lot of cases, what happens in the graduate, lab, where people, from PhD level and onwards are hired, is that people look at the quality of your research. So that might include participation in research, in papers, maybe recommendations from people who have worked with you before. But for an undergraduate, it's very difficult to get to that level. You probably have limited participation in research. You probably don't have a lot of references to share about your ability to work in a research environment. You probably had minimal opportunities to conduct research from beginning to the end and even know about all of the different stages that go into that kind of research. And a huge challenge for that is access to high quality lab environments. So as an undergraduate student, you have to go through a lot of training and potentially be in a well-funded institution to have access to a lot of that kind of equipment. And that's why bioinformatics is such a critical opportunity for those who want to develop those kinds of skills and who want to stand out in terms of their capabilities when they apply for research positions, when they apply for positions in the industry, or even when they want to continue their research and study in a university. So I want to briefly speak about the research fellowship program that we have that brings the opportunity to work with big data coming from expert labs, interact with mentors from around the world that have the background and the kind of track record and could potentially give you a recommendation letter at the end, and also the training that we provide, which will really highlight the different opportunities in both research and in industry on how bioinformatics is actually used. Okay, thank you. So those are the three main components of this program, right? And so I'll briefly kind of show how these three different components are used in several different directions that I think are very promising in the world of bioinformatics, where really you can see a lot of data coming to the forefront of biological discovery and translational research that leads into biotechnology, pharmaceutical industry, and the clinical research field. So there are kind of four of these topics that I will briefly talk about: precision medicine, population health, biotechnology and agriculture, and space and astrobiology. These four have been chosen because the amount of data, the complexity of data, and the reliance on computational biology skills, individuals, infrastructure, etc., is growing at an exponential rate. So let's first talk about precision medicine. What we all know is, when we think about the word medicine, is that at the beginning of that journey in medicine, you meet a doctor or a nurse. And as you probably all know, when you go to a doctor, you go to a clinic, the first thing that they're going to do is they're going to try to diagnose the disease. And diagnosing the disease is a process that involves expertise, so a lot of experience that doctors have dealing with this specific type of disease. It requires labs where your samples are being sent to identify what's going on. What exactly is this? And then it also has this large field of research that constantly is being updated, because people are finding new meaningful insights about different types of diseases and conditions that we call preclinical conditions that lead to diseases. So in this field of precision medicine, this whole change from having a few indicators that can tell us very accurately what the disease is, that is changing to many, many more indicators that are much more precise. That's where the word precision comes from in the term precision medicine. But also, because they are so precise, they are specific to very small groups or even individuals. And that's where the word personalized medicine comes from. So those two parallel directions of precision and personalized medicine converge in specific chronic diseases. And those could be cancer, metabolic diseases, and others. And they include a combination of various markers that can help us understand the risk, how predisposed someone is to have a disease happen early on, complexity of disease progression, so how likely are they to recover fast or develop some complications, but also importantly, to two very specific and immediate needs in this field, which is accurate diagnosis and then effective treatment selection, because it's important to select treatment that actually is going to be effective. So in the world of precision medicine, bioinformatics and biomedical analytics play a huge role, because so much data at every interaction is being collected that someone needs to make sense of that data. And that someone today are very specialized genetic counselors and maybe clinicians, doctors, But ultimately, this field continues to develop. And in between those interactions, there's going to be a lot of opportunity to, number one, conduct research that leads to identification of these markers, and also to technicians that will have to deal with this kind of data, process this data, analyze this data, and be able to say something about this data. So I think this field has a huge potential for those of you that are interested in medical school, that are interested in MD, PhD programs, but also that are interested in research that has the potential to change the way that people are diagnosed and treated for precision medicine. The second area that is very obviously right now being changed in front of our eyes is the world of public health or population health, where we see this pandemic and others really change our reliance on molecular diagnostics to identify specific strains, to identify populations at more risk, to understand new approaches to vaccine design, right? We're seeing it in front of our eyes. And so I think it's obvious that in this field, there's going to be a tremendous change as we move forward in terms of how much data and how much analysis of that data is being done to really be able to anticipate, react more fast to these kinds of outbreaks, but also to be able to design new therapeutics and interventions like vaccines to slow the transmission of diseases. Another important area is the field of biotechnology and agriculture. We kind of combine them together, but really I want to first talk about agriculture. So as you know, with climate change, there's a huge concern about the ability of crops to remain resilient. With the recent increase in the prices of fertilizers, we see a lot of people turning to bacterial type fertilizers that change the microbiome of the soil. But because land is so different, right? There's different types of toxicity, acidity, amount of water being available in different fields. Now we understand that we can effectively measure those different kinds of indicators and identify the best crops for the best portion of land. We can increase productivity of those crops, and we can make those crops more nutritious, more resilient, and easier to grow. And so the field of precision agriculture is really emerging as a field that will be dominated by data, but importantly also by omics data. You can see the number of crops that their genomes have been sequenced to be able to ask these questions is growing every year. And you can see that the ability to design new crops and make specific features of those crops become more attuned to the needs of a specific region. Those kinds of studies are emerging. You can see a huge issue with forests around the world, where they are being attacked by insects, and they're being subjected to harsh climate conditions. And obviously all of that ties into the quality of the environment that we live in. Finally, I want to briefly mention space and astrobiology. So when we think about space, we don't typically think about medical needs. We don't think about quality of life, right? I mean, astronauts go there and they train for years to be even considered. But eventually when they're there, they have to deal with these very harsh conditions. And those include conditions of microgravity, conditions of radiation, really limited food supply and diversity of food. So there's really a very unique kind of combination of factors that we know affect human health in a profound way. And that's why there's a huge effort in not only building technology to get us to space, but also developing methodologies to understand how space travel will affect health and how we can adapt to these conditions to be able to grow food, maybe on the space station. Maybe, as you probably have seen, the first Arabidopsis plant has recently been grown in moon soil here on Earth, but it was in moon soil, and that is a significant study that shows that it is possible to grow food on the moon, theoretically. But also there have been studies about the microbiome because there's an issue with different types of microbes kind of living in the space station, International Space Station, right? So there's a huge interest and funding available for those kinds of studies. And in fact, many of our interns and fellows have been able to conduct research studies using data that NASA is actually providing free of charge to the community at large, kind of encouraging students, researchers to be asking questions about what's happening in space and how can we understand and better prepare for the type of space travel that is envisioned by these space agencies. So to start addressing the variety of topics that we just discussed, I think Dr. Raghavendran already provided you with a clear framework on how to be able to address some of those questions. But I want to review again a couple of the slides that he already shared, but maybe focus on them in a little bit of a different fashion. So the first thing that he mentioned was the challenge with selecting a topic. We just saw 4 major domains, a lot of potential directions, and a lot of interesting questions that could be asked. And I think as you get to know every one of these domains and you are able to study the research publications out there that come out or have come out in the recent years, you will find that a lot of those papers, they focus on organizing and categorizing different topics relevant to that domain. So if you take the field of precision medicine or population health or biotechnology and agriculture or space omics, you will find that a lot of those kinds of experiments and studies that have made a significant impact on that field will be categorized. They will say, this is what has been the topic, this is why, these are the types of experiments that have been done to address that topic or question. The second thing is that in the discussion portion of those publications, you will find a lot of times the reviewers and the authors who write these papers, they will actually identify gaps, unanswered questions, controversies, and debates around specific topics. And those are really the most interesting topics that you potentially as a student or as a young researcher can start to think about. A lot of those are limited by the amount of data that is available. A lot of those are limited by the types of experiments that are possible to conduct. But this field is developing very rapidly. And so it is easy to imagine how a publication can only focus on a small portion of an unanswered question, and the rest of the data that is available from the experiment has yet to be analyzed in a specific way to be able to contribute to those answers. So I think that bioinformatics and the availability of this public domain data really allows us to leverage these high quality, expensive data sets that are now publicly available to be able to ask very specific, impactful questions that we can actually answer. And I think that this research fellowship program is designed specifically around that kind of need. It is designed to help you identify those, understand the context of where the question is being asked, find those data sets, and then be able to conduct analysis to be able to answer those kinds of questions. And so I think another part of this process is really an understanding of where you're coming from, right? And this is something that comes with experience. And this is also something that comes in all areas of research, right? So think about this process as something that you potentially will experience in a research lab, preparing to conduct impactful research. In all of these fields, you have to understand the terminology, you have to understand the different data types, how those data types are generated, and how they are processed and analyzed. After that, you have to be able to select a topic, not only that is interesting, but that you and a team can actually complete in a defined period of time. A lot of research projects last decades, and the objective of this program is not to get you into one of those projects. The objective of this 3 months or 6 months program is really to give you the opportunity to ask a question and be able to answer it with some kind of analysis in a quick 3 month or 6 months period. Now, you don't have 3 months and 6 months just to do that. You have to understand that it will take you some time to get prepared, to identify that question, and then to conduct the analysis, you might have only a month or so, or maybe a month and a half. So it really has to be planned well and executed with an ability to understand what the results will look like and will mean. And so that's why we also have this practical component here as well. So to support this process, we have a community that will actually be able to help you along the way, guide you to the right kinds of resources, and be able to help you to conduct the analysis and plan it in such a way that you will be able to complete it in a short time. I'm going to skip my next slide here that talks about the practical kind of alignment, but I want to highlight two specific outcomes for this kind of training here in the United States. So the first example really comes from a researcher who comes more from a biology and biochemistry background, Dr. Olga Udartseva. Now she works for a company, a pharmaceutical company, but when she joined the program, she was a part of a research lab at the Roswell Park Comprehensive Cancer Center. And her background was in cancer, and she understood that a lot of these studies in cancer, what the biologist's role a lot of times is to generate the data, right? Ask the question, maybe generate the data, and then receive the results and be able to interpret them. And she understood that that gap between conducting the research and giving over the data and then getting it back, right? That analysis part of it is really the gap that she can master on her own to get a lot more results out of it. And because she was able to understand the approach, the logic of the analysis, and acquire some practical skills, she had two very specific outcomes. And those outcomes, number one, is that she was able to use bioinformatics in her ongoing research. And number two, acquire those skills and certify that she has the practical ability to use those skills to advance in her career. So those are two very practical outcomes. The second example is from a researcher here at the Louisiana State University. She was a PhD student at the time, Thao Vo, and she actually was able to identify the specific types of data that she might be using in her ongoing research. And actually that opened a whole new opportunity for her to be able to pursue this in a more serious way. So early in her PhD, she was able to try out a lot of different tools, right? As you saw already displayed, we have a lot of different pipelines that are focused on the logic of the analysis. And in a short period of time, you might not have the time to really learn a lot of the technical skills. But if you understand the logic, you can then take that logic and decide, I want to now become an expert in this field, an expert in the field of transcriptomics or metagenomics or genomics or some other direction, and really develop a whole kind of skill set around that. And so that was really the outcome for her in terms of her ability to define that interest, to invest the time to really master a lot of those skills, and also for the career development to really be able to then showcase those skills and the way that they have been practically applied. So those are just a couple of examples. And now, finally, I want to finish with our upcoming research symposium. So we already had conducted 2 large events. We had over 1,000 people who have registered for each one of them. And we had great panels of speakers from both industry and academia, showcasing advances in bioinformatics and biomedical informatics, and talking about the opportunities for students in terms of their research and career as they complete these kinds of training programs. And so what we have discovered is that it is very meaningful for researchers and students from around the world to be able to present their research and get feedback from experts, which comes not only as a good or bad, but really focuses on the specifics of the research and with a lot of feedback. And so that opportunity to both highlight your research, get feedback from experts, being able to network and interact with them is tremendously meaningful for people that would like to continue or even begin in this career and really understand what their options are and where this could lead to. So in terms of this summer research fellowship, the objective of this program is to help you develop and complete a project throughout the summer and potentially participate in this research symposium, networking with the people in industry and academia, getting feedback, and maybe identifying what that next step might be. A really critical component that a lot of undergraduate students do not have the option to do. And so this is a unique opportunity to get started and actually get it done in the short time that we have during the summer. So with that, I'll pass it back to Sri Gauri, and hope to see you all in this program that's coming up.
Right. Thank you so much, Mr. Ilya Bronski. Now we'll be launching a poll to better understand our participants. So if you could take a moment to answer the questions. The first is, what is your educational or research background? Then the second question is, which specialization track are you interested to work on? And finally, which skill set you want to get hands-on? So if you could just take a moment to answer these questions. All right, so let's move on to the next part of the webinar. Next up, we have Mr. Mohit Mazumdar. He's the business development head at Pine Biotech. He has over 12 years of research and industry experience and having a PhD in computational biology and chemistry from Jawaharlal Nehru University, India. He is also an expert in machine learning applications in biomedical data and chemical data. He is currently heading the global business development for Pine Biotech USA and contributing to bioinformatics research and education by collaborating with universities in India and Africa and mentoring participants from various programs by providing bioinformatics research support, applying statistical and algorithmic approaches. He will be talking about the research fellowship university relations and outcomes. Over to you.
All right. Thank you, Sri Gauri. And thank you, everyone, including Ilya and Dr. Raghavendran, for giving us a clear insight of how this research fellowship program works. So today, I wanted to discuss with you or meet with you all to know you better, and also to give you the right kind of resources that would be relevant to all of you. So based on your expectation and the information that you have got to join this meeting, we shared this poll so that we can understand that where your interest lies, and then we can help you guide through this research fellowship program. So I have a really long experience working in this field of computational biology. I did my PhD, and before that, I did my master's in bioinformatics. And I was able to work on several of these projects and completing many of these and publishing a lot of them. So I think in here as a mentor, one of our responsibility is to complete those analysis that we plan, that we work through. So you saw the structure in this meeting. I wanted to quickly take you through a couple of things before we all move to the very presentations where we have the research fellows, student research fellows who will be presenting their work quickly as we are running out of time. So before we move there, I just wanted to ask everyone, do you have these similar questions in your mind? If so, then we will be happy to answer some of these questions in the chat and help you understand how this works. So I'll read them out, the questions that we have, that we get from the students all the time. So the entire research fellowship program, can this program be a requirement for the university internship? Yes, and I'll show you quickly the structure. What is the difference between 3 months and 6 month program? So in a 3 months program, we cover most of the things that you saw in this presentation, including how to work on a project and to complete a project. But within a 6 months time, because these projects, they vary a lot based on the research question. They vary a lot based on the group that is working on. It also varies based on the amount of time that you are giving to this research fellowship program. So based on all the time requirements and the flexibility that we offer, this could be done in 3 months, 6 months, and we usually have this option given to students when they have exams to pause the research fellowship and then start it again after their examination. And about the hourly commitment, so how many hours commitment does the research fellowship program requires? So it's again a question that kind of is personalized. It depends on the number of hours that is available for the student and the mentor. But usually the way it works is that initially, when you're going through the coursework and understanding that how all of these components work, you have to give at least 3 to 4 hours a day to understand how the coursework is aligned with the T-BioInfo platform and the example projects that everyone has to learn to be able to work on a research project. Followed by that, it is more about finding the right question, doing the analysis where the time can be managed according to your own schedule. So what are the mentorship options? So we have a group of mentors from industry, from academia, from our own company. That includes all of us who are present in this meeting. So based on the question, we can assign or we can suggest mentors who are expert in this specific field. Okay, the next question, can we choose any topic? Yes, that requires you to go through the coursework and understand what are the different areas of research, what kind of research is ongoing, and if you can find specific publications and prepare the project form that we discussed, then you can work on a topic provided there is enough data for you to work on. So the next question, can we participate in group projects? Yes, there's a great opportunity as we are collaborating with industry, as we are collaborating with academia, research labs. There are many projects that we are working on, which are cutting edge, which requires a lot of workflows where you can participate and work on those group projects. They are also part of the research fellowship program, but it is recommended that we provide that sort of opportunities for people who have already at least completed one project. The next question, can we publish our project? One of the major goal of this research fellowship program is to be able to highlight this research that you have done in your own profile. So publishing your project gives you that tremendous amount of exposure and makes you a part of the scientific community. So yes. And then the next question, the 8th question, is about pricing, payment option, and scholarship. So this research fellowship program includes several of these resources that we talked about. That includes the example projects, that includes the basic beginner coursework, that includes the T-BioInfo platform server access, that includes the access to mentors, group sessions, and this is an entire process that develops your project. So yes, there is some pricing options that are available for anyone to join the research fellowship program, but it is also advised advisable to understand this, that all of this payment is going for the resources that you have been provided with. Help students from low income countries, for students who have financial difficulties in doing a program, we provide scholarships from our end, and also from universities whom we have collaborated and offering these programs in together. So furthermore, if you have any more questions, I will request all of you to put it in the chat box, and our team will be happy to assist you, and we will be reaching out to you specifically to answer any questions that you have about the specific topics that we discussed, and also these questions that we have put together. So now moving on, I just wanted to discuss about our university collaborations and how all of these resources are helping a university develop student capacity, as well as develop research projects, as well as develop grants and publication. So to discuss about, we have already discussed about all of these topics in detail. So we have beginner coursework for students who are really new to this field, who would want to start their journey and would want to work on a project eventually. So we have a detailed coursework that enables anyone. So most of our students who are coming from the background, which is mostly biotechnology, microbiology, even computer science. So this coursework is built for biologists. It's meant for biologists to learn data science, to learn biomedical data science, to learn bioinformatics and apply to a real project. So there is a beginner coursework which is very detailed for anyone without a background to learn and apply this. Then we have these specific coursework on multi-omics fields, big data bioinformatics, where you will learn that how do you see these data, how do you visualize such a big amount of data and make meaningful information, get meaningful information from there by using several of these tools, including the T-BioInfo multi-omics platform. That helps, again, the beginners and anyone to get that infrastructure, run experiments, analyze data from a very easy to use point and click system. The one of the purpose of making this as a simple system is that the logic of analysis, the meaning of the research can be understood. And that is one of the main focus of this research fellowship training, this research fellowship program. And we are really happy with the outcomes that students are getting from this, because they're understanding why they're doing it, and then they're performing the research. So followed by that, we have this different various specialization, the kind of research that excites everyone that we have in this OmicsLogic research, where you can work on specialization fields such as space omics, and then you can work on very difficult problems such as cancer. And for all of this, I mean, after doing the basic training, the research training, you start doing your exploratory analysis and do your literature review, and followed by that data analysis and interpretation. So that's the training part, and that is well discussed in this meeting, and I hope that it's kind of clear to all of you. If not, we will be again sharing with you the resources that will help you understand. So the technical training, we discussed this. There are several of these data types that we are using: patient for clinical data, animal models where these experiments have been tested. Many of the drugs have been tested, which starts from a cell line experiments eventually. And then we understand, analyze, process, and annotate and integrate this sort of data for many purposes, such as drug discovery, which is a very important thing that you learn from this precision medicine talk today. And by the help of tools such as the T-BioInfo platform and coding languages such as Python and R, we are being able to do downstream analysis, which is really good for all of that things that we want to highlight, like a poster presentation, like the presentation that you do for your university or for a grant. So that's the technical training part. Followed by that, we have the introductory coursework, then the example projects that you have seen. Then we have the specialization training, and then you work on a research projects with very defined steps. So because of all of this, I mean, that's the reason we have been able to do it challenging. So all of this is well structured, there is a help within every step. There is support within every step for students from different background, from different educational and geographical location. Now, as you have learned about the entire process, like this entire process of understanding and writing, all of this is also supported by faculty projects. So once you have gone through these steps, we align your interest with the faculty project with the partner university that we have, and we have faculties who have proposed projects whom we are working with and are more focused towards the translational outcome, more focused about getting publications and getting that recognition. So these are some of the opportunities that we provide to students to work with faculty who have these defined projects and who can help further giving you the right kind of feedback to develop your project. So that is what I wanted to show you all in this short presentation that I have put together. But before I leave and pass it to Sri Gauri to help students come and discuss about their research, I wanted to tell you about a couple of opportunities. So we have campus ambassador opportunities and also scholarship opportunities and internship opportunities. These are for students who are well versed with OmicsLogic and would want to represent OmicsLogic in their campuses, or would want to work with us within our team to help awareness of bioinformatics. So these are some of the opportunities, again, for the participants who are in the US and who are in Louisiana area, and also those who are connected with the NEPI network. We are offering many of these training programs starting from June to the students who will be pre-registering, and we'll be reaching out to help them register for these fully sponsored programs where the students are studying in these universities. And also we are providing 50% and above scholarships for students who are coming from outside and would want to be a part of these programs, these training programs, which is more of to give you an understanding of how the different data types can be analyzed for a specific specialization. And also, like, the research fellowship program is being offered in the US, and if you are in the US, you can avail that program. If you are in India and if you are in Africa, you can avail the program and details that we are sharing with you in this meeting. So that's about it. And I would like to pass this back to Sri Gauri to invite quickly students. And we have very less time, like we have only 14 minutes left in this meeting. So we will just conclude accordingly and have a few presentations, and rest we will be able to send you through video recording. Thank you so much.
All right. Next, we'll have the much awaited research fellows presenting their research projects. First up, we have Ms. Huyen Pham. She will be presenting her research project on spike glycoprotein sequences of SARS-CoV-2 analysis, variant analysis. Over to you, Ms. Pham.
Hello, everyone. So I'm going to present on COVID-19 phylogenetic analysis. The project that I did at T-BioInfo for research fellows. So I will skip the introductions and go directly to the material. So the variant sequence collect from NCBI virus, including several variants of concerns for analysis. Then I use the ProjectBased platform to analyze multiple sequence alignments and generate the phylogenetic tree. So overall of the process is here. this is the picture of the pipeline, and here is the results. So this would enable me to observe the evolutionary relationship between sequence, and to look at it further, I also downloaded the multiple sequence alignment file, and here was something I found. This will include the positions that is less conservative, insertions, deletion regions. Yeah, so here is the zoom out of those that have high variations. After that, I map it into the protein download from the PDB. And then I found some results that most of the paper just mentions about the substitutions mutations related to the increase in viral adaptiveness. So from the study, I find some new thing that is the deletions, insertions regions. So I suggest that it might be due to the imperfections during recombinations, because the virus have high recombination rate. So this could facilitate some deletions and insertions at a particular site. So yeah, so that is the overall of my research. So there's also some limitations, that is some insertions and deletions regions that make the multiple sequence alignments slightly off the check. So I hope that it may be improved in the futures, so that we have a better alignment at some site. So in conclusions, so understanding about the viral rate and type of the mutations can help us to understand about the virus, the evolution of the virus, and get the best strategies for preventions. It also can help us to find some sites that have potentials of mutations in the futures and predict the structure, so that we have best strategy to keep with the high mutation rate of the virus. Yep, so that is the end of my presentation.
Thank you so much, Rae. You can also view rest of the research fellow projects on the OmicsLogic portal. Next, we will discuss about the research fellowship registration, and it will be discussed by Ms. Sonalika Ray. She is the community manager. Over to you, Ms. Sonalika.
Thank you so much, Shrigari. I would like to share my screen. Just a second. If any questions, please put them in the chat box, and I'll be shortly taking you through the registration process. So today we discussed about the research fellowship program, the structure of the program, and the different specialization tracks that are being covered. And also we heard it from Huyen. We also have more research project presentations that you can explore on the OmicsLogic Learn portal. So now those who are interested to get themselves registered for the research fellowship program, where do they go and proceed their application from? So this is the OmicsLogic Learn portal that you all saw, and you need to click on the research button over here. This will redirect you to the research fellowship registration page. Now, once you are on this page, I'll just share the link in the chat box. Yeah. So now once you are here on this page, you need to click on the Get Started button, which will give you a pop-up listing the fellowship amount for beginner and advanced level for 3 months and 6 months. If you are a researcher who wants to explore data from the big data repositories and work on your analysis and complete your project, then the beginner level works the best for you. But if you are working in a lab generating your own data set and you want to explore your own data set, then taking the advanced level will be more helpful. So the cost for the beginner level for 3 months is $225, and for the advanced level is $297. And for the 6 months of subscription, you double the amount. So now, why do we have a fee structure for the research fellowship program? It's because you get the access to the OmicsLogic research resources, the training resources, and also the T-BioInfo server that Dr. Raghav and Ilya talked about, where you get 5 terabyte of space. And also you get allocated to these research fellowship batch meetings, where you are able to take your queries from the mentor and get your questions clarified. And now talk about the research scholarship opportunities that you can leverage. So those who come from bachelor's and master's, the students who come from various fields of life sciences from bachelor's and master's, and they feel the need to get a scholarship, they can definitely fill out the scholarship form. So I'm going to share with you the scholarship form, and also you'll be able to fill out the form and reach out to Ms. Sparsh Thar. She's our business development associate, and she'll be able to assist you with your scholarship. So once you fill out the scholarship form, we'll review your application and get back to you. Now, to fill your scholarship, you need to visit the page here, scroll down, and you'll get the registration form towards the end of the scholarship page. After you fill out the scholarship application, we'll review your application and get back to you. Most probably, we'll also schedule a meeting with you and listen to your research interest and educational background. And also, if you have a research proposal, then it would be great. So that was very briefly what I discussed about how you can get yourself registered for the research fellowship program and get started with your independent project journey. If you have any questions, you can put them in the chat box, and also you can reach out to us at marketing@omicslogic.com. Thank you so much, Sparsh, for sharing the details. And also Sparsh has shared our Indian contact number, where you can reach out to her and she'll help you with your enrollment process. Also, we provide scholarships to our partner university students. So those who are from Amity University, Noida, Jaipur, and Chandigarh group of colleges, please feel free to reach out to us. And also other students who are seeking a scholarship, please feel free to fill out the scholarship form. I'll put the scholarship form once again in the chat box for you. And also you can reach out to Sparsh on our mail address for any assistance. That was it for today, and thank you everyone for joining today's meet.